Thank you, Jamie, for the introduction, and I would like to thank also the organizers for the opportunity to present today our work. So I'm just gonna get this. So I'm gonna talk about the work that we've done about an octopus peptide. I'm just gonna minimize that one. That it was found from the transcriptome of the southern sand octopus from Brian Fry and his colleague. And what was interesting about this peptide, it's small, it's linear, and it has this domain that is very characteristic of tachykinin or neurokinin peptides, which they're highlighted the relevant part, the amino acids in red. So these tachykinin peptides, usually they're binding to G-coupled protein receptors, and they are involved in neuroinflammation, pain, and more recently in cancer. So Yanni from King's Group he used NMR, and she found out that this peptide is unstructural and disorder in water. And these results were confirmed also by Andreas from Paul Alwood's lab, that he did CD, and he also showed that in the presence of 50% TFE, which is a lipid mimicking SOL1, this peptide adopts an alpha helical structure, which implies in a way that maybe this peptide is interacting with the lipids, or better with the phospholipids, that they are present in the membranes of the cells. So our interest in our lab is about finding venom-derived compounds that they're targeting melanoma of BRAF mutation, the dominant form of this cancer. So we tested this peptide in a melanoma cell line, as well as in a healthy neonatal forensic fibroblast cells. And we observed that our peptide was more specific towards the melanoma cells. We confirmed these results also in other BRAF melanoma cells. And we tested some other variants as well as a comparison to ours that Brian had previously found in the transcriptome from other marine species. And we were able to show that and we observed basically that our peptide was the most potent. So because this peptide, in an effort to make it more stable and enhance its deproliferative capacity towards melanoma cells, Patrick from Paul Alwood's group, he replaced methionine that it has at the end that basically is responsible for the oxidation of the peptide with neuroleucin, as well as, the acid became amidated, which is more specific and typical of tachykinin peptides, and then the negatively charged asparagine with pyroglutamic acid. And then when we tested the peptide back in our melanoma cells and compared it with a native, we were able to see that even though this peptide maintained the dose concentration dependency towards melanoma cells, it was less potent than the native peptide. So to understand better, maybe the metabolic needs or the energetic demands that these two peptides have, and they cause in the cancer cells, we use the seahorse flux analyzer, which basically that we originally used in a lab, and it allows us to show if there are any metabolic changes caused either by glycolytic parameters or by changes in the mitochondrial energetic machinery. So we saw that there were no differences in regards to their glycolytic parameters, and I just highlighted two as an example between those two peptides, and that is usually measured by extracellular acidification rate. However, it seems that the octopus PEP1, our peptide, seems to be more, had to have a higher basal respiration, maximal respiratory capacity, as well as a non-mitochondrial respiration needs in comparison to its analog. There were no changes in the ATP-linked respiration, which probably shows that maybe there is an inefficient mitochondrial oxidative phosphorylation coupling occurring here. So the octopus peptide, we were also showed that it has an increase, it causes an increase, a generation of the reactive oxygen species, Well, this increase of ROS was not accompanied by any changes in the mitochondria membrane potential of the cells. Also, at the time point we checked it, after 24 hours, there were no changes in the cell cycle phases, and the cells were happy. They didn't lead to any cell death. So Javier, my postdoc, was able to dig in a little bit further, and he interestingly observed that if you combine this peptide with an inhibitor of the complex 1,3 of the mitochondria, then it seems that this peptide enhanced its potential, but he observed no changes when he combined it with inhibitors of the ATP synthesis or the OX-phosphate. In order also to understand more where this generation of ROS is coming up, if it's coming from mitochondria or from another compartment or from the whole cell, he used the different and specific ROS scavengers. And he found out more that when he combined it with NAC, which is more of a scavenger, of a ROS scavenger of the total of cellular input, that's probably more likely the ROS is coming. It didn't look at this stage that is coming from the mitochondria, which is very interesting, and we are following this up to verify this observation. The other part we were really interested in, and we are, is what's the link? Is there any association between the neurokinin receptors and the observed antiproliferative profile in the melanoma cells? So Professor Glenn Boyle from QI Marburg-Hofer, he was able to show by using Affymetrix array assay that all of the tested melanoma cells, they were including, our model that we characterize more, our peptide, express at different levels, all the 3 different neurokinin receptors that I'm going to refer to them as neurokinin receptor 1, 2, and 3. So Javier, what he did is he set up this experiment, and he was able to check whether if you treat, the cells, the melanoma or the healthy cells with the octopus peptide, that there were no changes in the mRNA or in the protein level of the cells, showing that maybe there isn't any direct correlation between the observed antiproliferative profile. So then the next step was Lotten and Asa from Lewis group. They set up this flipper, the fluorimetric plate reader assays, which is basically they transfected the mammalian cells with the genes belonging to the neurokinin receptors, and using the appropriate pharmacological tools, they were able to show that the octopus PEP1 is a weak and, as you can see, an unspecific NK receptor agonist. However, the octopus peptide analog that Patrick had synthesized with just the 3 amino acids different, it seems to be more, as you can see here, more potent and more specific towards NK1 and NK3 receptor. And I forgot to mention in my previous slide that the focus was more on NK1 receptor, because this is, according to the literature, the receptor that is involved more in cancer, in the proliferation of cancerous cells, of tumor cells. And the fact that our peptides, the 2 peptides, were actually targeting NK1 receptor was also further demonstrated by Lotten and Asa, that they show that if they used the specific NK1 receptor antagonist, they were able to block the agonistic effects of the 2 peptides. Excuse me. So the next step, what we did is Gonzalo from our institute, he performed some molecular modeling, trying to determine if there was some specific structural characteristics, features, or belonging to the octopus PEP1 in comparison to the variants and the analogs, or that might help us to design later better analogs and explain its antiproliferative capacity. So by doing molecular modeling, he was able to show, and this is just an example, that our peptide seems to fluctuate between an alpha helix and a polyproline helix conformation. So this raised some questions. Basically, we wanted to see which is the active part. Is there a specific part of the structure, the N terminus or the C terminus, that is responsible for the activity, for the cytotoxicity of this peptide. Also, if you can see from the structure, it looks like it's got a lot of negatively charged amino acids. So if you replace these amino acids with alanine, which is a hydrophobic also residue, does this have an effect on the cytotoxicity? Similarly, if you change the location of these charges, like the geographical sort of features, does this have any effect? And as well as we were interested to see if we disrupt the binding of our peptide with the neurokinin receptor, what would be the result, the significance, as well as the significance of the polyproline conformation that it seems to be due to leucine, that it is one amino acid before the end. So replace leucine with third leucine. And then we created these analogs and we test them back in the melanoma and in the healthy cells, and they have a similar pattern, more or less. And we were able to determine that the active part is the C-terminus, that the hydrophobicity plays an important role, because when we substituted with alanine, it seems that it maintained its activity. The N-terminus was not important, and neither was changing the location of the charged amino acid. However, when we disrupt the binding, in theory, with the neurokinin receptors, and we make it also less hydrophobic, with a less hydrophobic residues, then we saw that, as well as the polyproline conformation, the peptide had totally lost its activity. It wasn't active at all. So the final test to understand, is there really a link between the neurokinin receptor and the cytotoxicity, was done by Sergey from Neely's group, who he actually created using the CRISPR-Cas9 technology, 2 pools of melanoma cells, that is just shown here, but they have knocked out the NK1 receptor by approximately 30%, similar levels, both of these pools. And then when we tested the active peptides in the wild type, in the control, as well as in the knockout cells, we didn't see any differences, suggesting that the activity of this tachykinin peptide is independent of the NK1 receptor. So the next step was we verified the antiproliferative capacity, the proliferative profile of our peptide in vivo, first by using the nude immunosuppressive mice, which we treated them for a period of around 2 weeks. Every 2 days, we did IP injections with the octopus peptide, and we were able at the end to determine that the tumor progression volume of the treated mice was less in comparison to the vehicle treated mice, and this is work done by Manuel. Similarly, these results were complemented by the zebrafish xenograft embryos, which was done this work in Laura's lab from the University of Compostela, which basically what Pablo and Fernanda did, they labeled the melanoma cells with DIL fluorescence, and then they injected in the tail, and then it went to the circulation of the zebrafish embryos, and then they treated the embryos for a period of 3 days. And then at the end, when they quantified the fluorescence, they were able to show that the octopus treated zebrafish embryos, they had a less melanoma cells in comparison to the vehicle treated. So just a very sum up slides, what we were able to show, that we have an octopus peptide that is targeting the melanoma BRAF cells. The active part is the C-terminal, which is also and plays a role hydrophobicity, the alpha helix, as well as this polyproline conformation that it has. This activity is independent of the NK1 receptor. And also these results were confirmed, it has a therapeutic potential, because we were able to show that in 2 different animal models, in the mice and in the zebrafish embryos. So just very quickly, this is the work I started when I was at John Miles' lab at QIM Marburg-Kopfer with the collaborators. And then Javier took over and he followed up and he did most of the part of this work. Manuel did the animals, Gonzalo, the molecular, the computational work. Laura's lab was done, the zebrafish work, animal model, and of course from the University of Queensland, all the collaborators that they either provided the peptides and the variants, or they did the NMR, the CD, and the synthesize as well, the peptides.